Okay, hello, welcome to the Musella Foundation Brain Tumor Webinar Series. Tonight's special guest speaker is Mike Ciesielski, who is the CEO and co-founder of MimiVax. Dr. Ciesielski's background is a PhD in molecular immunology and has 25 years experience in neuro-oncology research. He started his career with Roswell Park Comprehensive Cancer Center in Buffalo, New York, where he first invented the Survaxm vaccine. Since then, he has built MimiVax as a spin-out company to develop Survaxm with the goal to allow him to bring this vaccine therapy to GBM patients. Go ahead, Mike.
Great, thank you, Al. So thank you for giving me the opportunity to talk tonight on the webinar series. And we greatly appreciate all the work that you do with virtual trials. And it's really an honor to be able to present some of our work and share it with the brain tumor community and patients that have also tuned in tonight. We're doing our best to bring new therapies forward to help patients, and we really want to be able to provide them hope that there is something coming, and hopefully we have something already that we can share with them and hope to take care of this dreaded disease. So a little background on what we are. MimiVax is a small company, small pharmaceutical. There was actually a spin-off of Roswell Park, as Al mentioned. We've really brought this from the laboratory that I started with. This is something that I discovered, invented in the lab, and we built the company around it with the thought that somebody has to do something to get more options available for brain tumor patients. And this is what we focused on and thought was the best promise in our lab, and have been really marching forward ever since to try and get it closer and closer to FDA approval. So I put my disclosure up. I am a co-founder and shareholder of MimiVax, as well as co-patent holder. So obviously the great unmet medical need, as many of us are very unfortunately aware of, is that glioblastoma does not have many treatment options. The median overall survival is only 16 months. If you look at the NCCN treatment guidelines, the first thing you see is consider clinical trials. And I think that is horrible. I think as a society, we have to do better. 93% of patients don't get to experience a 5-year survival rate. We clearly have to do better. So what I'd like to share a little bit of data around is our Survaxm cancer vaccine for glioblastoma. This recently completed a phase 2A single-arm clinical trial in newly diagnosed GBM patients. And we're moving forward from there into a phase 2B clinical trial, which is currently open and ongoing and recruiting. Survaxm, as I mentioned, is a cancer vaccine. It's a type of immunotherapy. In our phase 2A, to cut to the chase, the overall survival that we observed was 25.9 months, and the progression-free survival was 11.4 months. So we think we've started to make a dent in what would be observed for GBM. As a company, our pipeline is centered around Survaxm, and I bring this up just to show that there are several trials ongoing with it. As I mentioned, the first study and our main indication is newly diagnosed glioblastoma, which is now in the phase 2B randomized study. There was a recurrent study with a combination of Keytruda that is just completed in collaboration with Merck, so we're anxiously awaiting data from that. It's in data analysis. There is a multiple myeloma study that was in obviously a different indication, blood cancer, and see how this would apply to other forms of cancer. There is a metastatic endocrine tumor study that is open right now. It's neuroendocrine tumors, which, not to be confused, isn't necessarily a brain tumor, but it's more of a metastatic abdominal tumor that arises from neural type tissue. That's running at Roswell Park. And we also have recently opened a pediatric high-grade glioma study. And this is opened in collaboration with the Pediatric Brain Tumor Consortium and actively recruiting patients with high-grade glioma, relapsed medulloblastoma, ependymoma, and now opening an arm for DIPG. So we're excited to get that off and moving. And then other things in the lab, we're looking at an antibody that's derived from the vaccine and a CAR T cell. These are preclinical studies looking to learn more about how the vaccine works and what else we can do with it. So Survaxm targets a protein that's expressed in brain tumor cells, and it's a protein that's overexpressed. It's abundant in these cells. That protein gives tumor cells the ability to live longer than they're supposed to, aptly named Survivin. This is something that prevents tumor cells from undergoing what they call programmed cell death, prevents them from dying and going through that cycle. It's highly expressed in a number of cancers, not just GBM, but as well as renal cancer and liver cancer and melanoma. It's expressed in almost 90% of cancer. So there's a hope one day that this can even move beyond GBM. It's not normally expressed in adult normal tissues. It's a type of what they call a fetal oncoprotein. So it's a protein that's part of fetal development and then usually turned off shortly after birth or early childhood. And the rest is a little technical there, but there are multiple forms of Survivin. So when I mention Survivin as a protein, I'm actually referring to 7 different proteins that are located in and around the cell. Some are inside the nucleus, the inner portion of the cell, the cytoplasm, the next portion out, as well as some that's out on the surface of the cell. And that's important for the vaccine that can actually target it through an antibody. So Survaxm is an off-the-shelf immunotherapeutic. It's a non-cellular based vaccine. There's no patient materials involved in it. It's kept in pharmacy as a freeze-dried powder in a vial, ready to go whenever a patient is identified. It's a peptide-based conjugate, and what that means is that it's a synthetic machine-made peptide that's attached to a protein. In this case, it's a large protein called keyhole limpet hemocyanin. This is a protein that's harvested from the blood of a sea snail off the coast of California, and it's very strong to provoke an immune response. And then it carries the peptide, which is a mimic of the Survivin structure. What that means is that it's not the Survivin peptide, but it's structurally similar to the Survivin molecule and triggers and trains the immune system to create an immune response that can then recognize that wild type Survivin. It's given as a subcutaneous injection. It's delivered in the arm, much like you'd get the flu or COVID vaccine. Patients receive 4 doses every 2 weeks, and then hopefully every 2 months as a booster, as long as their tumor does not recur. And then this produces a T cell and an antibody response that targets those multiple forms of Survivin in and around the cell through different mechanisms. So it's targeting through CD8 cells, which are cytotoxic T cells, CD4 T cells, which are helper T cells, as well as antibody. This is just a schematic of how that works. It's injected, it's picked up by antigen-presenting cells. These are macrophages, dendritic cells. They eat the protein up and then present it to stimulate the immune system, in this case, triggering the CD8 cells to replicate, as well as triggering some antibody production. Both those T cells and antibody converge on the tumor cell, leading to an attack that leads through surface recognition of Survivin protein by the antibodies, as well as T cell engagement through what's called MHC-associated epitope antigen presentation, more of a traditional T cell mechanism. So this is some data from the phase 2A study. This was recently published in the Journal of Clinical Oncology. This is a single-arm, multi-center study, 63 newly diagnosed GBM patients from 2015 to 2020. This was performed at Roswell Park, Cleveland Clinic, Dana-Farber Cancer Institute, Mass General, and Beth Israel Deaconess Medical Center in Boston. These are all adult patients over 18 years of age. All patients had tumor resection with minimal residual enhancement. The vaccine was then delivered again subcutaneously as an injection. It's delivered in a emulsion of montanide, and this is basically an oily carrier form that allows the vaccine to be slowly eaten up by the immune system. And and then it is also given with a side dose of sargramostim or GM-CSF. This is a cytokine that stimulates dendritic cells and leads to a little better antigen presentation, which fires up the immune system a little better than without. The schematic down below is how patients would receive therapy. So all patients, again, have resection, go through chemoradiation, and then there's the 28-day period between chemoradiation and the start of temozolomide adjuvant therapy, where patients are recovering from radiation resection. This is a prime area to inject the vaccine. These are patients that have a little less immunosuppression. There's no other therapies going on at that time, and we're able to grab hold of a little better immune system and prime that immune system a little better than coming in a little later in the study. And that was fairly successful. So they got 4 doses every 2 weeks, and then again, those boosters every 2 to 3 months thereafter. The population of patients is shown on the right, and this is generally what you'd expect for a newly diagnosed population. We didn't really see anything special about that. MGMT status, IDH1 status. Survivin expression was from about 1 to almost 40% in some cases. Because it's an immunotherapy and survivin is what they call a cycling protein, it means that all those cells will eventually express survivin. So we just need to make sure some cells are expressing. If you take them out and grow them in a dish, you see that all the cells eventually turn on survivin. And that's what happens in the patient as well. And this is the data. So this is the overall survival. So the light blue arms on the bar graph to the left represent patients who have not progressed. Darker blue is when they have progressed but are still with us. And the gray bars are those patients that have passed on. And what we learned from that was that overall survival increased to 11.4 months and median overall survival increased to 25.9 months, but importantly with a range going all the way out to 78 months. And what's very interesting to us, and I guess to many, would be that of those 63 patients, 16 of those patients are still alive over 56 months, and 9 of them have not even progressed. So we found that there is a group of patients that do very well with the vaccine and continue to do very well with the vaccine. And this is something that we're actively researching to try and find out what characteristics there are with this group that makes them possibly more sensitive, and if there's a way that we can better target them in the future. If you try to compare to other studies, we just point out so that it's 25 months from the first dose of vaccine, as well as 28 months from diagnosis. So roughly the first dose comes about 3 months into the treatment plan of a newly diagnosed patient. This is just comparing to some external controls to put it in perspective what the survival means. The last 5 large phase 3s that have not really moved the needle average about 15, 16 months overall survival. We know that. And we came in 25.9 months overall survival. If we stratify for MGMT methylation, so those are the patients that are sensitive to Temodar and patients that are resistant to Temodar, we see that the overall survival for those methylated patients shoots all the way up to 41.4 months. That makes us very excited because that would normally only be about 24, 25 months for that population. And we do see some activity in those unmethylated, hard to treat patients that, as we know, really don't have many other options. In this case, even a 2.5 month increase in survival is truly significant. So we're trying to find out what more can be learned from this group. Longitudinally, we see that at 5 years, 27% of patients are still alive, whereas we'd only expect to see about 5% of patients still alive under standard of care. And 30% of patients on trial survived greater than 50 months. How the vaccine works is we've monitored through immunomonitoring. In this case, we're looking at T cells. So we can see patients before they receive vaccine and then after vaccine. We can track those T cells and how they respond to the vaccine. We see that they produce cytokines and markers for maturity, interferon, things like that that are important for those T cells to function. At this time, they didn't correlate to overall survival, so we're trying to figure out how the T cells fit into the mix of things. But what did correlate to overall survival and correlate very tightly was the antibody response. So in our patients, after they, so there's a baseline sample, and then they get their first 4 doses, and then their boosters, they all produce a very strong antibody response. That's this IgG. And what we noticed was those patients that produced a high titer of IgG were also the patients that did extremely well. In fact, these patients averaged about 41 months overall survival for the entire population. And that was statistically significant, P01. It was very exciting to see that come out of the trial. And then if we stratify to various other molecular markers for GBM, we saw that those high titer patients were associated with enhanced survival with every different stratification that we did on the study. So the IgG was certainly important. This just looks at it another way on a standard Kaplan-Meier curve. So we see patients with high titer survive 43 months, patients with low titer 15, but it also held up for progression-free survival. Those patients that had the highest titer also did a little bit better overall progression-free survival. And again, that was statistically significant. Safety was very good. It's a peptide vaccine. We don't see a lot of toxicity from things like that. The most common reaction was an injection site reactions, pain, swelling, redness, injection site reactions. The highest adverse event we saw was a body rash, and that resolved. There was no SAEs observed on the study. Now we're opening we're not opening, but we're in a phase 2B study, which is a continuation of this study, but also adding a randomized aspect to it. So to get through FDA approval, one of the things that we do have to address is there has to be a control arm for the study. We wish there didn't have to be, but this is how the FDA works right now. So it is randomized 3-2 to give a better chance of being in the study arm. So it's patients that receive vaccine or they receive a saline component of vaccine, but they still receive standard of care. They still receive temozolomide. So it's a comparison of vaccine plus standard of care. This is now open in 11 centers across the country. Again, Roswell Park, Dana-Farber, as well as newcomers such as Atlantic Health, Overlook Medical Center, Fred Hutch, UCSF, NYU, Miami, and Northwell. This is looking for endpoints of primary endpoint of overall survival, secondary endpoints of progression-free survival, as well as biomarker and immune response analysis. So this is the quick summary of what we're doing. Survaxm is a new cancer vaccine. It's data-driven, published in Journal of Clinical Oncology. We think we have a unique mechanism of action through the antibody response, 25 months overall survival, which is pretty incredible, I think. Median progression-free survival, 11 months, 27% 5-year survival. And then those long-term survivors that are all approaching 5-6 years at this point. Some of them doing very, very well. Some of this was highlighted on NBC News a couple of weeks ago, where they did a few pieces on us. This was really exciting to get that out to the brain tumor community and hopefully get this available to more and more patients. And just a quick rundown of who we are. Primarily, our company is about 5 or 6 people. We're a small group, but we're shooting for the stars here. This started with myself and Robert Fenstermaker, the neurosurgeon at Roswell Park. We formed the company and have been moving through to clinical trials ever since. Some of the standouts for our advisory board were Manmeet Ahluwalia, was very out there in the neuro-oncology community. Elad Levy, as well as past CEOs of Roswell Park and CEO of Delaware North, Jerry Jacobs, have been a strong benefactor to our work to date. And just some references. I'm sure we can put those up somewhere so you can get access to them through the website or through the slide deck, which I'm sure will be part of the presentation. And at that point, I would like to end that and open it up if there's any questions or discussion or how this moves from here. I'll turn it over to Al.
Okay. 1 second. Okay, that was excellent. Very impressive. And I'm very glad that you came and gave us this presentation tonight. There's lots of questions. First question is, why would methylated or unmethylated MGMT matter for a vaccine?
So it's interesting. It's something we're looking at as well. I think there is a little bit of Temodar effect as an adjuvant. So I think Temodar can stir the immune system up a little bit, leads to some secondary responses. But what we do see is that the methylation aspect and the DNA damage aspect also seems to affect the immune system. So then there's more damage, we get a stronger immune response. It's unusual. We're trying to find out what the molecular characteristics are around that, but it's something that's still a work in progress.
Okay. Have you ever tried this in a person who hasn't had the tumor removed?
There's been a couple of early cases that we tried. And one of the things about immunotherapy is that it's going to be much better at cleaning up residual tumor than it is trying to affect a large bulk tumor. The vaccine is really good at preventing recurrence. That's how we like to think of that. So it does affect a bulk tumor, but the effect is not very as robust as we would like to see. It's much better at preventing those recurrences.
Okay. Why not give the first shot before the surgery? Why wait till after radiation? Because once you get radiation, you're gonna have a little bit of immune problems.
Yeah, so it's an interesting paradigm. For us, we've been trying to piggyback this on the back of standard of care without having to reinvent the wheel too much. But it is a thought that it'd be very interesting to see a neoantigen, neoadjuvant phase of the vaccine. So it's something that we're looking at, but it's not something we've done yet. Okay.
Is there any possibility of getting expanded access of this vaccine?
Not at the moment. So basically with our size and having to reserve enough doses of the vaccine for patients on the phase 2B study, we're not able to offer it outside of the study at this time. We hope that changes, but at the moment, no.
And you don't have a trial for recurrent GBM right now, do you?
Not at the moment. So that one just closed up. So that was the recurrent plus Keytruda study. So that one is now in data analysis. Depending on what we learn there, we can get another one open up shortly, but that's a ways off.
The Keytruda help much? You don't have the results yet, do you?
We don't have the data yet, so I don't know what to say there.
Theoretically, it should though, right?
That was the idea. So there was, Keytruda takes the foot off the brakes, and we give the vaccine as a shot of gas in the tank to see how best that will respond. So in theory, it should be great, but you always have to wait for the data.
Okay. Do you allow multifocal glioblastomas?
No. So it's only single site, single focus.
And does Optune disqualify you?
We did exclude Optune for this study just to keep it consistent with our phase 2A study, which was done prior to Optune becoming approved. So we're really trying to recapitulate what we saw in phase 2A and the 2B as our fastest way to get that to FDA approval without muddying the water too much.
You're not going to, well, the question is, what is the best timing of using a checkpoint inhibitor along with Survaxm? That's a tough one.
Don't know yet. I would suspect that we would want to do that afterwards. It's one of the things we do see as patients progress. So we do follow the immune response. And a number of patients that have progressed early or even later on, we have noticed that there is an immune cell exhaustion phenomenon that starts up. So it would make sense that Keytruda would help combat that. But like I said, we don't know yet.
Can you compare your vaccine to the other types of vaccines, such as DCVax?
Sure. So ours is one that's non-cellular based. So it's no patient materials, pulled out of pharmacy. Ours is specifically tuned to target survivin and its family members. So that gives us the ability to have a sharp focus on what that antigen response is going to look like. One of the things that we've noticed as we've developed this vaccine and others is that if you have a broad spread of antigens, sometimes that's good, sometimes it's not, because you have what are called immunodominant antigens. Your immune response only picks up on certain ones. In some cases, those are good. We've observed in some cases that there are antigens that can actually be detrimental to the immune system. So we always have to be careful of that. The other vaccines that are out there, though, such as DZFax, is exciting because we're seeing other forms of immunotherapy that are getting into the brain. And I think there's plenty of room for all these vaccines to treat patients and possibly even combine someday. So it's really a great thing to see any of these vaccines getting off the ground.
Okay, that was the next question, if it made sense to combine vaccines. So you answered that already. What is the path to FDA approval?
So right now we're trying to convert the phase 2B actually into a pivotal study. So we're hoping that as we come up to an interim analysis of this, which should be sometime late next year, we can approach the FDA to allow us to convert the phase 2B into a pivotal study. We're in the process of getting our CMC and peptide manufacture up to the point that will be considered for pivotal studies. Right now it's designed to switch over to pivotal from the get-go. So it's part of the plan.
So what would the best case scenario be for FDA approval?
Best case scenario would be an accelerated approval based on the interim analysis. Probably a long shot, but we always try for that.
Data like that, I don't see how you know.
You never know. Yep. So if that's not the case, then we would convert into pivotal status. We would have to recruit about another 270 patients to double to increase statistics and continue for, I would assume, another 1.5 to 2 years of follow-up. And then we'd come to our finalized endpoint data and present that for approval. So hopefully it's all part of the same trial is what we're trying to do here.
Okay, if a patient has a tumor, but it shrinks to less than 1 centimeter from the radiation and surgery, are they ineligible for the trial?
As long as it's less than 1 centimeter, yeah, they should be eligible.
Oh, wait, it has to be less than or greater than?
It has to be less than.
Oh,
less than 1 centimeter. Yeah, less than 1 centimeter residual tumor, whether that's through surgery, radiation, or some part of standard of care. It's designed to be given in combination with standard of care, so that's what we're really trying to compare here.
How difficult is it to manufacture in large quantities?
So it's a peptide vaccine. So the peptide aspect is fairly easy. You can make a lot of that. The other side of it is a protein that's harvested from sea snails. So there is some limitations to how much can be produced from that. We think that we should be able to have enough to treat plenty of patients upon approval, but it is a biologically sourced component of the vaccine. So
would it be possible to make an mRNA version?
It is. It's something that we're also working on. We don't know if there's a true advantage to the mRNA aside from the manufacture aspect of it. Yeah. So what we've seen though is that some of the mRNAs aren't as strong of an immune response as you would get from some of these peptide combinations. So, if you think about the sea snail that we're using, it's almost like inducing a shellfish allergy in a patient. So you're really trying to induce something nasty for the immune system to recognize. And so far, it seems to be working pretty well.
One of the slides showed a breakdown by haplotype. Did you notice any blood types that responded better?
No. So this was something that was actually fairly interesting to us, and that's why I put it on there. I'm actually glad somebody caught that. So when we first started, we were looking for this as purely a T-cell vaccine, and it was restricted to HLA-A02 and A03 patients to get an idea of how that would work. As we learned more from the studies and realized that actually the antibody is the main thing, we started to get away from the HLA restriction and started expanding the studies. So the next study included HLA-A11 and A24. Eventually, we saw there really wasn't a difference to the T-cell responses, that because it's a long peptide, which means that there's 15 amino acids in this peptide, and an immune response typically responds to 9 of them, which means that many different patients can pick different pieces of the peptide to respond to. Long story short, we found that the HLA restriction didn't have any practical meaning for the study, and we dropped it from the more recent studies. So this is no longer part of the phase 2B, but we didn't see anything different in any of the haplotypes. Everything that we've looked at so far has generated a response. It hasn't been a deciding factor, so that's why we got rid of it.
Okay. Is MimiVax a public company?
No, we're still a small private company. So we're actively raising funds to be able to continue phase 2B and ramp up our production. One of the goals there is to be able to acquire enough resources so we could open this up to expanded access. I mean, that's one of our limitations, that we are a small group. This is a company of 5-6 individuals and as a spin-off of a major cancer institute. So we've been dependent on donations, angel investors, family offices, and quite a few donations that have come in from the Buffalo community through bike rides and fundraisers have gone into supporting many of the patients on the study.
We had the same problem with a pediatric drug called Onco-1. And what we actually did is the brain tumor community got together and funded expanded access program. We actually ran it also. Would you be open to something like that?
Yeah, absolutely. Anything that we can do to get it out there to patients faster has really been what our goal is. It's sometimes frustrating having to go through the established pathways that are there by the FDA. I mean, you understand why they're there, but at the same time, I think there's other ways to help patients.
Oh, that leads right into my next question. What do you think about the Promising Pathway Act?
This is something I'm really excited about. And as you probably got from my last answer, that it's a kind of a pet peeve of mine that, drugs take so long to be developed. And, here we are in a brain cancer study, a brain cancer drug, where we all know there's 1 or 2 other drugs that are available. There's no options for patients, and that's just simply not fair to anybody. And I think if there's drugs that are out there that show a signal of efficacy, that are safe, and that aren't going to bankrupt families, I think that's something that's important to me too, because there's always that tendency that some unscrupulous companies could take advantage of that and charge an awful lot to get their drugs. And we don't want to do that. We want to be able to make it available to as many patients as possible and make it fair to everybody. And I think the Promising Pathways is designed for that. I think it's a perfect opportunity, perfect disease for this.
Perfect.
It just makes sense.
Okay, combine 2 of these questions. You require less than 1 centimeter of residual tumor. Do you have any reason to believe it's not going to work with greater than 1 centimeter or with multifocal tumors? Or it's just not going to work as well, or it's not going to work?
So the bigger the tumor, there's always this push-pull going on with the immune response. I think the immune response can only handle so much of a tumor. And a lot of that's based on previous studies. There's been a little bit that we've learned from some early compassionate use patients that have all pointed to a smaller tumor, less residual disease is ideally what you want to do. I mean, that doesn't mean that we can't try this in the future, but at the same time, there's only so many scenarios that we can look at in these trials. So we've tried to focus on what's worked so far and keep expanding from there.
Are you looking for more than the 11 sites that you currently have, or it's going to be limited to 11?
Right now, we're limiting it at 11, just because enrollment has been extremely well from that group. It was actually a lot more than we expected. So we're good right now.
We sent a lot of patients here. It's one of my favorites.
Thank you.
Let's see. As far as the control group goes, was there any thought or discussion with the FDA about using external controls instead of randomization to placebo?
Yes, we looked at that briefly. FDA would not accept any external control plan that we presented to them. What they had offered was that we had to look at patient-level external controls. What that means is that every patient on study would have to be matched with a patient from a database. And as we pursued the databases that were out there, which is kind of surprising to us, there's not enough data to match these individual patients.
We currently have enough data for a good external control group, and we're doing it for 2 other clinical trials right now. It's too late for this, but keep that in mind in the future.
Yeah, no, that'd be exciting to try.
Survaxm cures to have a large regulatory grade registry with a lot of glioblastoma patients, a lot of other types of cancer patients too.
Great news.
Let's see. There's no reason to think it's not going to help with recurrence. It's just you're trying for newly diagnosed, correct?
Yeah, so we've had some experience with recurrence. So our first phase one was a small study, 10 patients, and we did see an overall survival of about 15.8 months out of that group just receiving the vaccine. There were a couple of long-term survivors in there. Actually, I shouldn't say a couple because most of the patients on study lived over a year. One of the things that was interesting there is that we started that as a first-in-man toxicity study with patients that had tried just about everything. Were on their second and third recurrence, and all of them survived a year, which surprised us. So we quickly rewrote the trial to include the booster dosing. That's where that came from for the later studies. And we managed to carry some of those patients out to 7 years with 2 and 3 times recurrent GBM, which was very exciting. But we did decide that the better spot right now is the newly diagnosed. And we're hopeful on the recurrent plus Keytruda study to see what that shows us.
Okay, that's about all the questions that we had. I want to thank you so much. This was excellent. And like I said, this is one of my favorite trials, and we keep sending patients to you. And
Thank you.
Talk about trying to get an expanded access program going.
Sounds like a good plan. I appreciate everybody coming on and appreciate the time to talk about the vaccine, something that's near and dear to our hearts. And we wish everybody well and good hope for the future.
I'm going to post the video of this on the website tomorrow so you can see all the links. Thank you and good night.
Great. Thank you very much.